going to introduce our third speaker, which is my co-host, Shou-Wen. He's going to give us the third talk. Shou-Wen received his bachelor's degree in engineering physics from Tsinghua University in 2013. So we heard this university before. And our first speaker apparently got his bachelor's degree 3 years prior. Shou-Wen went on to obtain a PhD in physics from the same university, Tsinghua, in 2018. During 2018 to 2022, so the next 4 years, he worked as a postdoctoral researcher in Department of Systems Biology in Harvard University, Harvard Medical School, and he was awarded the Quantitative Biology Award from the Damon Rennan Cancer Research Foundation during his postdoc. In 2023, he joined Westlake University as assistant professor in the School of Life Sciences. I think I remember that a couple months after he joined Westlake, I was there visiting the university, and that's the first time we met. So Shou-Wen had a joint appointment in the physics department at the School of Science. Shou-Wen pioneered the field of lineage tracing and developed several cutting-edge technologies, including MethylTree, the first non-invasive lineage tracing tool based on DNA methylation epi mutations, which opens the door for single-cell multi-omic lineage tracing in humans. So Shou-Wen, please take it away. Thank you.
Thank you. So can you see my screen?
Yes.
Great. It's my great pleasure to speak here. So I will talk about how to infer cell lineages with evolving DNA methylome. In 1983, John Soston used live imaging and obtained the entire cell division histories in C. elegans. Using this, they discovered apoptosis, which was awarded with Nobel Prize in 2002. Therefore, fundamental discoveries could be made by just looking at the cell division history. So what can we learn from the lineage history? We can learn whether a cell divides or not, and what kind of cell types it can differentiate into, and whether it can migrate to different tissues. So these are very useful information when studying embryo development or cancer metastasis or hematopoiesis. To study cell division histories, we can label cells and with distinct and heritable markers, and then analyze these cells later to see which are from the same founder cells. This kind of labeling technique can be very powerful. For example, Barker and colleagues used such labeling approach to prove that LGR5 positive cells can give rise to all the downstream cell types in intestine. And this provides the definite proof that LGR5 positive cells are intestine stem cells. However, such a labeling approach requires genetic engineering, which is not applicable to human studies. In humans, we can utilize the somatic mutations accumulated during cell division to infer cell lineages. However, these mutations are extremely rare and therefore very, very hard to detect. The mutations in mitochondrial DNA is an alternative source for lineage tracing. However, each cell has hundreds of mitochondrial DNA, and the inheritance of mitochondrial mutations suffer from neutral drift and selection. Indeed, its reliability has been questioned in several recent papers. Therefore, we have to look for alternative endogenous markers for lineage tracing in humans. Maybe we can consider epigenetic markers such as chromatin accessibility, transcription, and DNA methylation. But which one should we use? Our recent work actually provides important insights. Together with Fernando Camargo and his former postdoc Lily from Westlake University, we developed Darling, a powerful lineage tracing mouse with extremely large barcoding diversity and highly efficient barcode labeling and recovery. Additionally, we developed Chameleon-seq to jointly profile gene expression, chromatin accessibility, DNA methylation, and also lineage barcode from the same cell. And this work was published in Cell in 2023. And last month, we published a companion Nature Protocols paper to present specific experimental and analysis details when working with Darling mice. With both Darling and Chameleon-seq, we evaluated the clonal memory in different modalities. We first barcode the hematopoietic stem cells at E10, and then these cells migrate to feed the liver and then to bone marrow. And we collect these HSCs after 10 weeks and profile them with Camellia-seq. We visualize the similarity of these cells for each modality. And here, the color of the cells indicates the clonal origin. You can see that these cells do not share similar gene expression from the same clone or similar cloning accessibility, but the cells coming from the same clone do share very similar DNA methylation. This is even after 10 weeks of labeling. Therefore, DNA methylation maintains long-term clonal memory and should have the potential for lineage tracing. DNA methylation is a chemical modification on the DNA sequences. In humans, it happens mostly at the cytosine of the CPG site. A given CPG site can either be methylated or unmethylated without changing its underlying sequences. As the cell divides, its DNA methylation can be copied to its two daughter cells. However, in this process, it can make errors such that an unmethylated C becomes methylated, or vice versa. Such epi mutations on DNA methylation accumulate during cell division and could potentially be utilized to infer cell division histories. In fact, it has been previously estimated that the epi mutation rate is roughly 10 to minus 3 per CPG site per division. Because we have around 29 million CPG sites in human genome, this amounts to 10,000 CPG epi mutations per division. Furthermore, these epi mutations, once generated, can be stably maintained for hundreds of cell divisions. Therefore, DNA methylation epi mutations carry both rich and stable information for high-resolution lineage tracing. To build DNA methylation-based lineage tracing tool, we first need a method to profile DNA methylation in single cells. The classic approach is single-cell bisulfite sequencing. A more recent version is CABRNet, and both methods will convert the unmethylated C to T, and the unmethylated C will remain as a C. So we can sequence this data and to resolve which CPG sites are methylated. The next question is, can we infer cell division histories from the single-cell DNA methylation measurements? There are 4 computational challenges to be addressed. First, the single-cell DNA methylation data is highly sparse, typically with 95% of the missing data. And second, there are sometimes large technical variations between cells, which could skew the weak lineage signals. And third, it is well known that different cell types, they can have their own specific DNA methylation profiles, and it can be challenging to identify which methylation changes are functional and which are stochastic epi mutations that can be used for lineage tracing. And finally, during development, the global DNA methylation level undergoes large modulation, and which could erase the stochastic epi mutations accumulated previously. So in January of this year, we published a method called MethylTree in Nature Methods, and it successfully addressed all the 4 challenges. Below, I will explain this method in detail. Given the sparse single-cell DNA methylation data, we first compute a cell-by-cell similarity matrix and then use it to infer the lineage tree. To address the problem of massive missing values, we would compute the Pearson correlation between 2 cells using just the detected CPG sites in both cells. For example, to compute the similarity between cell 2 and 3, we only use these 3 shared CPG sites. And to compute the correlation between cell 5 and 6, we only use the least 2 shared CPG sites. To test this approach, we simulated a cell division process with epi mutations and generated a sparse single-cell DNA methylation data with just 5% genomic coverage. And applying MethylTree, we accurately reconstruct the entire division histories of this process. Next, we performed the benchmark with epi mutation with experiments. And here we isolated 4 single 293T cells and cultured them into 4 distinct clones. The last clone was further cultured to give rise to 2 subclones. And then we perform single-cell bicycle sequencing with 5% genomic coverage. Now, applying MethylTree, we computed the methylation similarity between all these cells. The color of each cell here represents the clonal origin of each individual cell. So the raw methylation similarity shows that cells coming from the same clone, for example, this green clone, are indeed tend to cluster together, but not entirely. For example, you can see that these blue cells seem to have higher similarity with these blue cells from the same clone, but unfortunately, they are not clustered together. Why that is the case? It turns out that our measurement has noise. The more noise a cell has, it will have less correlation with other cells. And importantly, different cells may have different noise strengths, and this could skew the result. So we developed an iterative approach to identify the noise strengths associated with each cell, and then corrected the similarity matrix. After this, the corrected similarity matrix shows perfect lineage structure, and even for the 2 subclones, P9 and P10, they are also clustered together. Therefore, MethylTree could infer lineage histories at 100% accuracy from sparse single-cell DNA methylation data. So far, we have successfully addressed the first 2 challenges of missing values and the technical noises. Now, let us fix the third problem that different cell types have their own methylation patterns that could interfere with lineage tracing. We first simulated a differentiation process where a stem cell could either become cell type A and B, each with distinct DNA methylation patterns. As before, we computed the methylation similarity between these cells. Here, the first column indicates the clone identity of each individual cell, and then the second column is the corresponding cell type identity. We can see that the similarity matrix is completely separated into by the cell type information. And we hypothesized that the row similarity matrix can be decomposed into a cell type signal matrix T and a lineage signal matrix L. And we developed a computational approach to regress out the cell type signal. And applying this approach, the resulting signal correctly shows the lineage history in this simulation data. To test this approach in real data, we turned to a public single-cell DNA methylation dataset. Here, we have 2 embryos, and each has both the fetal germ cells and the somatic cells. The question is, can we infer which cells come from the same embryo? In fact, the fetal germ cells have very, very low global DNA methylation level, while the somatic cells have high methylation level. Indeed, the row methylation similarity of these cells are separated by their cell types. However, we can see that within the fetal germ cells, there are 2 clusters that corresponding to 2 different embryos. Applying our method, cells are now perfectly separated by their embryonic origins. And this proves that our method works remarkably well, even with cell types having very different global DNA methylation levels. We sought to further benchmark MethylTree with more complex differentiation system with multiple different fate outcomes. And we took the blood progenitors from the mouse bone marrow and labeled these cells with synthetic barcodes so that we know their lineage origin. Then we cultured and differentiated these cells in vitro for 6 days and performed single-cell sequencing to get its transcriptome, DNA methylome, and the lineage barcode simultaneously within a single cell. We look at the transcriptome and found that indeed the cells are differentiated into many different cell types. Next, we computed the methylation similarity between these cells. Here, the first column is the ground truth clonal origin from our measurement, and then the second column is the cell type information of each cell from the transcriptomic data. And we can see that the methylation similarity are clustered into many different blocks, and each block corresponding to exactly 1 ground truth clone. So in fact, there are 52 clones with the ground truth labels, and they are all 100% identified by our MethylTree method. So we isolated the 21 clones with multiple different fate outcomes and generated the lineage tree based on the de-methylation similarity. And here the inner ring is colored by the ground truth clonal label, and the outer ring is colored by the cell type. We can see that here all the 21 clones are correctly separated in this lineage tree inferred from de-methylation data. How deep should we sequence the genome to get accurate lineage tracing? To address this question, we downsampled the coverage in each cell and found that MethylTree requires more than just 5% genomic coverage for nearly perfect lineage reconstruction. And since single-cell de-methylation data is very sparse, it's very common to average the data within a large genomic bin, like 100,000 base pair, in order to fix the problem of missing data. We found that at such a large genomic bin, the lineage accuracy is almost zero. In fact, for accurate lineage reconstruction, we have to use small bins, like, for example, 250 base pairs. This curve is obtained after remove the technical variation between the cells. If we do not remove the technical variations, you can see that the lineage accuracy is substantially lower. And it highlights that we have the importance of noise correction in lineage inference. The above results are from mouse blood. We also carried out a similar experiment using human blood cells. Here, we also observed 100% lineage reconstruction from single-cell de-methylation data, despite that cells are differentiated into different cell types. In early embryo development, cells may undergo drastic modulation of their global de-methylation level, and this could erase shared IP mutations between cells, therefore interfere with de-methylation-based lineage tracing. To test the feasibility of MethylTree in this context, we used public single-cell de-methylation data from full cell stage in mouse embryo, and we found that MethylTree correctly separates cells by their embryonic origins. We observed similar success across different stages in mouse embryo development and also in human embryos. Therefore, MethylTree works even with global modulation of the de-methylation profiles. We also tested MethylTree in the context of cancer. Here, we used the public single-cell de-methylation data for colorectal cancer cells. The inferred copy number variation can serve as a ground truth lineage label. And we infer the lineage tree from de-methylation, and it agrees very well with the copy number variation information and suggests that MethylTree also works for cancer cells. To summarize, we have carried out a comprehensive benchmark of MethylTree across different cell types, developmental stages, and also in different species, including mice and human. In all these cases, we observed nearly 100% lineage reconstruction based on single-cell de-methylation data, and this makes MethylTree a non-invasive and versatile lineage tracing tool across broad conditions. Also, we have successfully addressed all the 4 challenges to infer lineages from single-cell de-methylation data. In our first application, we studied the fate decision in human embryo development. Here, at the blastocyst stage, cells are partitioned into surfactant and inner cell mass, which would have different functional roles later. It's unclear when the first fate decision is made in embryo development, and this is partly due to the lack of high-resolution non-invasive lineage tracing methods, and we attempt to address this problem with MethylTree. So we computed the methylation similarity for all cells from a single human blastocyst embryo. Then we use it to reconstruct the lineage tree. Here, this represents the zygote and is the first cell division, and this is the second cell division. And this represents one of the full cell stage cell. And you can see that all the progenies from this full cell eventually become the inner cell mass. This suggests that the fate choice was made at the full cell stage, and we repeated the analysis for other human embryos and obtained similar results. So we concluded with a stochastic early commitment model for human embryos, whereas the full cell stage, one of the full cells are strongly committed to become the inner cell mass. Our view is also supported by several recent papers published at the same time. For example, using GFP injection method, Shou-Wen showed the existence of early fate bias in human embryo in 2024. Using advanced histone profiling method and also proteomics, people also identified the molecular heterogeneity between cells at the 2-cell stage, thus providing a possible molecular basis for early fate decision. Next, we apply MethylTree to study hematopoiesis in mice. There are thousands of hematopoietic stem cells, and they maintain the entire hematopoietic system with billions of mature cells. So how many distinct HSC clones exist in mouse blood? In our cell paper, we barcoded HSCs at E10, and when these HSCs just begin to form, and we profile these HSCs with chimeric in the adult stage. So we could simply apply MethylTree to reanalyze this dataset. In the methylation similarity matrix, we found that cells are separated into distinct clusters, and each corresponds to roughly one HSC clone generated at E10. Therefore, each methylation cluster corresponds to a de novo HSC at E10. And we could just count the number of methylation clusters to estimate the number of HSC clones. To doing so, we identified around 250 HSC clones, and the result is consistent across 3 mouse replicates, where the HSCs are profiled at different stages. One thing that is intriguing is why we would observe distinct methylation profile for each HSC clone seated at E10. Our hypothesis is that in the early embryo development, there is quick accumulation of epi mutations. Then a very small fraction of these cells are randomly sampled on a division tree to become HSCs. And due to this selection bottleneck, these selected cells have very different demethylation epi mutations. And these seeded HSCs will continue to expand with ongoing epi mutations, but the epigenetic bottleneck defines the distinct methylation signature between different HSC clones that we observe in our data. As cells divide, it naturally accumulates demethylation epi mutations. We just need to sequence the DNA methylome of these cells to obtain the cell division history in humans. Therefore, MethylTree opens the door for high-resolution, non-invasive lineage tracing directly in humans. And it can be used to study cell lineages in various contexts, such as human embryo development, cancer metastasis, and hematopoiesis. In our lab, we have established a cost-effective MethylTree platform for non-invasive lineage tracing in humans. Here, we can sort cells into a 96-well place, and for each cell, we can obtain both its transcriptome and the DNA methylome. Then, using MethylTree, we can infer the cell division history. Therefore, this platform simultaneously obtains the cell lineages, transcriptome, and the DNA methylome in single cells. And this single-cell multi-omic lineage tracing platform allows us to study human tissues with a relatively cheap cost of just $5 per cell. So far, we can use the mutation in mitochondrial DNA, nuclear DNA, or demethylation for lineage tracing in humans. As I mentioned in the introduction, the mitochondrial DNA has complex inheritance and do not accurately record cell lineages. Although the remaining 2 methods can accurately record cell lineages, the demethylation-based method has its advantage. Because in each cell division, a cell can only accumulate just around 1 somatic DNA mutation, but about 10,000 epi mutations on DNA methylation. And this makes a very big difference. Because it is very hard to detect the real somatic mutations. This approach is challenging to implement and costs $500 per cell and does not support single-cell multi-omic readout. In comparison, our epimutation-based approach is easy to implement, costs just $5 per cell, and support single-cell multi-omic readout. My lab currently has 8 members, divided equally between computation and experiment, and we focus on lineage tracing tool development and its application to specific biological problems, such as hematopoiesis and human embryo development. So the MethylTree method is developed by myself, and I was very fortunate to recruit my student Mengyang Chen in 2023 when I just started my lab, and he quickly established the wet lab and carried out all the benchmark experiments beautifully. And Rui Jiang also contributed a lot by analyzing a large amount of data. And we would not be able to finish the work so quickly without the experimental support from my colleague Lily at Westlake University. Finally, we are recruiting postdoc, PhD, and research assistant for both experiment and computation. And contact us if you are excited with either lineage tracing or DNA methylation, and I'm happy to take any questions.
Yeah, thank you so much, Shou-Wen. Yeah, here's a question. Does the methylation-based cell lineage tracing require the methylation profile of the ancestral cells? Does the method still work if all these cells are gone and only the fully differentiated cells are available?
So if the progenitors are gone and only fully differentiated cells there, then you can still recover the lineage relationships between these mature cells.
Okay, and the next question is, do extranuclear environmental factors affect expression and stem cell fate? This is a question that you can see on the side of
yeah, I can see,
the
yeah.
screen.
So yeah, I mean, that is certainly possible. Yeah, so here we are only exploring using DNA methylation for lineage tracing. And other factors like chromatin sensitivity and so on can certainly affect the gene expression and the stem cell fate outcome, yeah.
The next question is from Kun Zhang. How many cell divisions apart between two cells can MethylTree resolve at 100% accuracy?
Yeah, so we recently carried out another benchmark experiment using PF-1 cell lines, and we basically we take a single cell and then culture them and then sequence cells after a few days. We found that currently, at least we can resolve cell divisions after about 5 days. We haven't tried final resolution, but I think if we sequence very deeply, it's possible to have even better resolution.
And does MethylTree work better for some cell lineages than the other cell lineages, or for different developmental stages?
So
because of the DNA methylation, it's also a regulation of lineage commitment. So it may work differently in different lineages.
yeah, so that is possible, but from our experience, we also recently tried to apply MethylTree to just directly to human blood, where it has many different cell types, and it works very well. So different cell types or differentiation associated demethylation changes is not a real issue here. But it's possible that the MethylTree may not work so well for human brain because the cells there do not divide actively, and they can accumulate a lot of other changes in the DNA methylation.
I see. And the epi mutations remain stable over 1,000 cell divisions. And so I think the question is about epi mutation stability. Is there possible conditions where, or in some tissues, that this stability can break down, potentially limiting the method's applicability?
Yeah, this is a great question. So first of all, the estimation that the stability is based on a single experiment in cell lines, and that is also a result from a focus on a few locus in the genome, and we don't know very well the IP mutation rate in the genome wide. I mean, for example, in different locus, what is the IP mutation rate? So we don't know that yet. But back to the stability question. So I think because there are so many CPG sites in the human genome, even though some of them are not so stable, and there are so many signals, each division can accumulate 10,000 IP mutations. So even many of them are not so stable. So there are some stable signals, and that can help to reinforce the lineage information. So I don't think that is a big problem here.
And what are the next steps for improving MethylTree? For example, are there plans to identify genome regions that could further enhance lineage inference robustness, or to adapt the method for clinical applications?
Yeah, so currently we use the whole genome demethylation for lineage tracing, and this is costly. So I think if we are able to identify certain mutation hotspots for lineage tracing with demethylation, that will dramatically reduce the cost and make it more affordable for other people. Also, we are also trying to benchmark MethylTree in different tissues. And because our lab have the darling mice, so we have a unique advantage to do lineage tracing in vivo with mice. And so we have the ground truth lineage label, and also we can use demethylation to reconstruct the lineage. And we are in the process of comparing MethylTree with the darling to see how well it works across different tissues, different conditions. And that will also enable more people to use this tool in their own study.
All right.